All right, so let's go ahead and get started for today. Graded the exams, posted the exams. You know, some of you did well, some of you maybe not so well, like, not as well as you would have liked. And so I'd like to take a few minutes at the beginning here to talk about what you can do and again reiterate what I think are good study strategies for the class. So I'm working to get everything put up, a little bit ahead of time. The best strategy that I've found, and this is by asking people who do very well in the class, make sure that when you come in here and see the slides up here and me talking about them, that's not the first time you've seen it, right? So you don't have to read the book and be prepared to pass a test on it, but you should try to read the book or at least look through the figures and figure out what you understand and what you don't understand from that. So read through the assignment that we're going to be covering in class, and then as you get to the parts that you don't understand, and there will be some, don't freak out. Don't spend an hour trying to dig up the answers or whatever. Just write down. Have a piece of paper. Take notes on what you're reading. Have a piece of paper. Write down, man, here's exactly what I don't understand. I'm looking at this and this is what I don't get. Then you come here and your questions to ask me in class are already written out, unless the explanation that I give makes it so that you can understand and you're okay. You have your list, so you're not going on the fly from what you've read ahead of time, and you're like, oh, well, okay, so I still don't understand this part of dominant epistasis, which is what we're going to talk about, was what we're going to finish talking about today. And, he just talked about that, and I still don't understand, and here's my question that I've already got written out about exactly what I don't understand. Hand goes up, you can ask the question. Make a plan to go to the ISL or come to see me. My office hours are Tuesday, Thursday, from right after class gets out until about 12:40. Today they're going to end at 11:50 because I have a meeting to go to, and then our prep person for genetics is out of town today, so I'm having to take some time to make sure that everything gets set up there. But normally it's 10:40 to 12:40. Come and see me. A lot of people have good success becoming a regular at the ISL. If you don't have anything after this class is over, get on the stairs and walk up one floor and park yourself in the ISL. After you've read through things ahead of time, then you've heard the lecture, you've asked your questions if you have any, then go over it again and take some more notes and see if there's anything you still don't understand. And then there are very few times in the ISL that there's nobody there who can help you with genetics. There are, I think, 6 different people who can help you with genetics. All of them have taken the class. All of them have gotten either an A or a B in the class. And all of them, I've seen their name and said that they're good. So these are not, these are people who have passed the test, so to speak, and they should be pretty good to help you out. As you're going through all this stuff, I have problems assigned at the end of the chapter. Work through them as we're going through the chapter, right? Do not wait until even 1 week before the test, because it is too late then. Go through the problems as we cover the material in class. And I think I said this earlier, but if I didn't, the book is really good in the problem section where it says, like section 5.1, and here's 5.2, and there's 5.3. It says, it specifies which section of the chapter goes with which problems. Go ahead and do those as we're going. Something else to think about too. And so every semester, including this one, I have people not do well on a homework or a journal assignment or something else, and they're kind of flummoxed because they've paid Chegg or one of these other companies to check their answers, right? And what almost always happens in these cases is that the people who are checking your answers are not getting paid very much and they're going very fast, and what you have written is not incorrect, but it does not answer the question and they have not checked. So I am free in my office hours. If you cannot make office hours, send me an email with a few times that you're available. Mondays and Wednesdays I'm free almost all day, except that I'm usually in my lab and not in the office. So if you come by, I'm not likely to be there, but I am very willing to meet with you. Just we'll set up a time so that I know what, when you're coming and can make sure to be around. The ISL people, like I've said, they've got 6 genetics tutors up there, all very good, also free. Also, all of them know what assignments I have and know what I'm looking for because they've taken the class in the past. So I want to be as encouraging as possible to everybody, no matter what you got. This is one exam. You have 3 more exams. You have a bunch more journals. You have a bunch more exam homeworks. But if you didn't do as well as you would have liked, now is the time to get on it, and to get on it pretty good. The worst thing that you can do, and this happens because I hear about it all the time, is, oh my God, I didn't do very well, and you freak out and you just freeze and you stop working. And that's not going to work out for you. You've got to not be afraid of it. Every semester I have people coming in, and it's usually like around the 3rd exam, and they're like, oh man, they figured it out. And I said, well, this is the correct answer, and they're all surprised. Well, this is hard. It can't be right. I can't have figured this out. Yes, you can figure it out. Do the stuff I've talked about. Go up to the ISL. They will help you. They will help you with the homeworks. They will help you with the journals. They will talk you through them, but you can't go up, they might ask you to do some more reading or something, because you can't go up there not knowing anything. They'll try to talk you through everything, but it helps if you come like what I said, having looked through everything and knowing what the specific things that you have questions about are and why you don't understand something to the best of your ability. That is helps them to help you better, because if you're not doing well and they don't know where your understanding is breaking down, they can't just look at you and guess. All right? So let's go ahead and get on with the rest of chapter 5 here, and we're going to start where we left off, which is we're talking about dominant epistasis. So remember, epistasis is where what happens, the phenotype generated by the results of a cross at one locus, affects or masks the effects of what happens at a different locus. Here, it's dominant epistasis, so white color for the squash ends up being dominantly epistatic to the other locus. The second locus goes between yellow and green. So here's the way that it breaks down. When you're having an epistasis, remember, you get everything adds up to 16, but the ratios are just skewed. Here, the 9 plus 3 is 12, meaning anything with the big W will be epistatic or mask what happens with the Y. You can't show green and you can't show yellow. You'll be white only. But let's just knock this off right here, and let's just look at this part right there and the Y locus. You notice anything about that? It's just a monohybrid cross, right? If the white locus didn't exist, then you would just be yellow squash, 3 yellow squash and 1 green squash. Yellow is dominant over green. But white is epistatic to both colors. So only 1/4 of the squash have color, because with the epistatic locus, 3 out of 4 are big W. So there's 1 big W homozygote, 2 big W little w heterozygotes, and 1 little w homozygote. So only 1/4 of all the squash are able to show any color at all, and then from them you get another 3-to-1 split. Make sense? Let's look at what is happening here. So yeah, 1/4 of the non-squash are green. We already talked about that. So here is the pathway. So in many cases, when you have epistasis, this results from the 2 genes encoding enzymes that do jobs in the same biochemical pathway. Here you have compound A, which is colorless, getting converted by enzyme 1 to compound B, which is green, getting converted from by enzyme 2 to compound C, which is yellow. All right? So, whether you are a residential student or drive home every day from here, there's a very good chance that you cross more than one bridge on the way home. Can we have that as a given, right? So, you can think of each of these enzymes as bridges, and these are the compounds are like the land in between them. So if the first bridge you come to is out and you have to stop there, it doesn't matter whether every other bridge you cross on the way home is good or is also out. The first one is epistatic to the rest. Whether the first bridge is in or out determines whether any of the other ones matter. If you don't have enzyme 1, you don't make the white compound turn into the intermediate green compound. It doesn't matter if enzyme 2 is working or not, because enzyme 2 can only work on compound B, not compound A, the colorless one. Does that make sense? All right. And then if enzyme 1 is working, then it does matter here, and big Y will specify enzyme 2, converts green to yellow. If you have 2 little y's, enzyme 2 is not working. So what's going on here is big W either encodes a dominant mutation that makes the enzyme not work and poisons the reaction, even if you have the enzyme there, or big W could encode some kind of inhibitor of enzyme 1. So in either case, having one big W allele means enzyme 1 either doesn't work or isn't there or what have you. And so that's what stops things up. So it's dominant epistasis because it's a dominant allele that's having the epistatic effect. Remember last time we talked about recessive epistasis, where we if you had 2 little e alleles, then you couldn't get any fur color in the Labrador retriever. So recessive epistasis, dominant epistasis. Now we're going to talk about duplicate recessive epistasis. All right? So here we are going to cross 2 snails that are both have a white shell color, and in the F1 you get all pigmented snails. So pigmented must be dominant over non-pigmented, or the white color. And then if you cross these to each other, you would get a 9:7 phenotype ratio. What's 9 plus 7? 16. Because your classes add up to 16, what can you say? It's epistasis, not a lethal allele. There's nothing missing, they've just been rearranged. So what's your normal dihybrid cross phenotype ratio that you get? 9:3 to 3:1. So what is going on here? It's the last 3 that go together. 3 plus 3 plus 1 is 7. So you have to have one. We're just using A and B for the gene names. You have to have one dominant allele for both of those to get color. If you are recessive, for either gene A or for gene B, you show no color. So this is duplicate recessive epistasis because gene A, gene A is going to be epistatic over gene B, and gene B is going to be epistatic over gene A, right? So let's look at this pathway. This is what I was talking about last time when I said, well, whether you have codominance is just dependent on your point of view. In the last example, it was dominant epistasis because you had compound A, which was colorless, go to compound B, which was green, and go to compound C, which was yellow. Everything could tell the difference. In this pathway, compound A gets changed to compound B by enzyme 1 again, different enzyme 1. But compound B is also colorless. So you can't tell if the snails contain compound A or compound B by looking because you can't tell the difference. If you had a biochemical test for compound B, you know, some kind of a, you could put it in with a reagent and the reagent would change color if compound B is there. Then it would be A is epistatic to B. But here, because both A and B need to have one dominant allele in order to see color, you have duplicate recessive epistasis. Each gene can have an epistatic effect on the other one. Here you can just think of it as being as simple as gene A, dominant allele A, codes for enzyme 1. Dominant allele B codes for enzyme 2. Little a means that you make defective enzyme 1 and therefore you don't go from A to B. And little b means you don't make any good enzyme 2 and you can't go from B to C. Right? Make sense? All right. Recessive epistasis, dominant epistasis, duplicate recessive epistasis where you just have each of the two genes capable of being epistatic to the other one. And now we're going to go to duplicate interaction. This is not in your book, but I found some delightful pictures of summer squash. Summer's almost over, but hopefully you had some of this on the grill. They come in, they used to be like the long shape, right? The crookneck squash, or these little what are called patty pan squash that are, you know, like looking like little flying saucers. And also lately they've been, in some places in farmers markets, they've been selling in different colors these ones that are the intermediate shape, sometimes called 8-ball, or you know, they're just sort of like a round baseball shaped squash. So disc, sphere, and long. If you cross a disc to a disc squash, you will get all discs, so true breeding. If you cross a long to a long, you will get all long, so true breeding. Disc crossed to long, so true breeding disc to true breeding long, all disc F1. So what's that tell you? That disc is going to be dominant over the, you know, over the other ones. But there's also going to be epistasis going on. In the F2 generation, so this is just a dihybrid cross, these are all going to be big A, big B, little b, right? You will get 74 disc, 50 spherical, and 8 long. I'm not going to make you do the math. That ends up being 9 to 6 to 1. What's that mean? You got epistasis because you're counting them up and you get 16, but which two classes are combined now? The 3 and the 3. All right, so I don't have the answer. Well, there's a chart on the next slide, but I don't have the answer on the next slide. So let's think this one through. How does this work? So the F1 are all going to be big A, big B, little b, right? Okay, and they're all going to be this. Then in the F2, you're going to have 9, 6, 1. So let's do the 9 first. We know what that is. What's the genotype of the 9 going to be? Big A blank, big B blank, right? So at least one allele. What is the long going to be? It's the one where you get only one. What you're going to have for the A's and the B's, they all have to be lowercase, right? The 9 to 3 to 3 to 1. So you are a disc shape if you have at least one big allele for both A and B. You are long if you are homozygous recessive, little a, little b for both. Because there is no difference here between, as long as you have one big A and one big B, it doesn't matter if you're heterozygous or homozygous for both of those, right? So we know that. All right, let's look at these guys here. There's two classes of 3 and they're different. What's one of the 3 class genotype going to be? So 9, the 9 is big A blank, big B blank. What's one of the 3 classes going to be? Who said that? Yeah. So little a, and what else? What with the B's? Big B blank. So this one here is going to be the little a, little b. We're talking about these guys in the middle. So you're right. One of them is going to be little a, and then big B blank, right? What's the other one going to be? Yeah, big A blank, little b. Let me write this stuff up so we can. All right, so here's what we got, right? So this is what we figured out. It's 9 to 6 to 1. So what can you come up with as a guess for what the genes are doing here? So if you have a big A and a big B, like these up here at the top, you have at least one big A and at least one big B, you are the flattened out disc shape. If you have no dominant alleles, neither a big A or a big B, you are the long shape. These are the intermediate. So either this one or this one gives you that intermediate spherical shape. Yeah. No, tell me what they're just tell me what they're doing.
Yeah, so because you have the little b, that's making it long, and then because you have the capital A, you're combining the long with this, which makes this sphere. So you're doing it in both of them, the little a, and the capital B. You're just combining the traits together.
So being both little a and little b, you get no, let's call it flattening power, right? If you get a big A and a big B, you're completely flattened out, right? If you get a big A only or a big B only, you're kind of in the middle, right? So I think what you're getting at and what the answer is that each of these has a flattening power to it, I guess you would say, right? So down there at the bottom, you have no dominant alleles for flattening of shape. In the middle, you have one allele for flattening shape, and up at the top, you have two. So when you have two, you're really crunched out to the disc shape. And when you have only one, you're the round shape, but it doesn't matter if you have the A or the B because they both have a flattening type shape to them. So this is called duplicate interaction. And it's on the chart that I'll show, and it's on the next slide if you've already printed them out. But before I go to that, I want to give an example that we've talked about. Now we're only talking about what we're talking about here is, I'll leave that run. What we're talking about here is two genes. These are all like a dihybrid cross. But remember previously when we were talking about the pea plants, and I said Mendel he was lucky he had a completely dominant allele. So when you had the tall and the short, there were the tall pea plant and the short one, right? And I said if I lined all of you guys up on a floor that wasn't slanted, from tallest to shortest, it wouldn't be like tall, 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 boop, short, right? There would be a continuum the whole way down. And the reason is because our genes that specify height are similar to this. But there's not just two of them, there's at least 8 of them. And in that case, the interaction that happens is even more pronounced than we have here, because here, it doesn't matter if you are a heterozygote big A little a or big A, you're still going to get the same flattening power, so to speak. But in human height genes, every dominant allele, like for more height, adds up. So with 8 loci, that means you have 16 possible height alleles. Yes, someone's already thinking this. There's more than 16 possible heights. Well, all your other genetic, the rest of your genotype goes in with those as well, plus plus your environment and all that. But what I'm saying is there's like 16 different extra height possibilities. Here there's only 3 things. You can either have no alleles for flattening and be long, you can have 1 and be round, or you can have 2 and be that disc shape. Okay, so now let's go on to the next page and look at the nasty table here. So we've already talked about all these. So in a regular dihybrid cross, 9:3 to 3:1, this has no epistasis going on because you can tell the difference. Seed shape has no effect on color, and color has no effect on seed shape. Then we talked about the Labrador retrievers. That's 9:3 to 4. Recessive epistasis, having, in this case, and they're just using A and B for all of them, having one of the 2 genes be recessive will be epistatic over or mask whatever happens at the other one. Little e made you a yellow Labrador retriever, whether you have the genes for black fur or brown fur, because if you can't put any color into the hairs, doesn't matter if it was going to be black or going to be brown, it doesn't happen. Then we talked about dominant epistasis. It's the same as this, only it's a dominant allele. You don't have to be recessive. In our squash example, having a W, or in this case they're showing it as an A, having one big allele for one of the 2 genes is all it takes to go over anything else. So if you have a big A, you are one color regardless of whether you have a big B or not. right? The duplicate recessive we just talked about today, 9 to 7. This is where if you have one or the other here, recessive allele, they're both going to show epistasis to the other one. So if you have two little a's, doesn't matter whether you have big B or not, you're going to be white color for the snail. And if you have oops, here it is, two little b's, it doesn't matter if you have a big A or not. And then we just talked about the duplicate interaction where you have that intermediate, the two middle classes go together. There aren't any examples for these. I'm not going to go into those, but you can see by the chart, you know, if it's duplicate dominant, that means one dominant allele would overpower anything else, and the only way you would see another phenotype is if you don't have any dominant alleles at all. So, and this too, you know, you can figure it out. But I'm not going to ask any questions on either of these two. But you have to know the rest. The thing to do is to think of examples. Look at the examples in the book. Do the problems at the back of the book. I'll put one of these onto the homework or the exam homework or a journal assignment. Make sure that you can understand these. Do not take this chapter or take this table and try to memorize it, because I'm not going to just tell you get 12 to 3 to 1. What's that? And you burp up dominant epistasis and get the points, right? I'm going to give you an example like the color in squash, but it's not going to be that. It's going to be something else that works the same way. You should be able to think your way through these. Think of what goes together, like the Labrador retrievers here. If you have this, now knowing how all the different kinds of epistasis work, you should be able to figure it out. The two classes that go together is where the epistasis is happening, or in this case 3, right? The classes that don't combine is where the epistasis is not happening, so you should be able to figure it out. Look, here's a 3 and a 4, right? So what's the difference between these should be this one here should be different because it's got a big B blank and the last one has just a little b. But if these two are both having the same phenotype and the only difference between them is the big B blank and the little b, then you can see that having little a means that whatever is at the B locus is meaningless. Do you see how that works? This is how you win this game, right? Understand how it all goes together. Look at the examples and think of how it all works, like knowing about the biochemical pathways and all that stuff. All right. I've not got any questions yet today because this is maybe a little ooh, but you know, think of things and ask me questions. You can ask questions next time too if you want. All right, so now we're going to go on. Talk about genetic complementation. Complementation is not misspelled. Complement with an I is when you, truthfully or not, tell somebody that their hair looks nice today, right? Or when something is free. A complementary appetizer is with an I. Complement means a full set. I always ask this, and there just must not be the right area for it. Has anybody been in or have a relative who's in the Navy? No. The Navy just gets no love. So, what? The Navy. Oh, you got, yeah, okay. So, if I talking about, because it's the only ship I know a number for. If I talk about an aircraft carrier has 5,000 sailors on it, have you ever heard that as termed like the ship's complement? Right? It's the total it's the total amount. So complementation test is the total that you're going to get, and it's used. It's a cross that's used to tell if two alleles that have the same phenotype are alleles of the same gene or if they're alleles of different genes. All right, so here's two individuals, and we're only showing one chromosome. Mutant A and mutant B both have the same mutant phenotype. You can see that they're at the same place on the chromosome. I'm telling you, this is an example, I'm giving you that these are both alleles of the same gene, the A allele and the B allele. So wild type maybe is like tall plants, and these are both short, but they're different mutations in the same gene. They're different changes, but just both happen to have the same phenotype. Perform a complementation test by crossing homozygote, or true breeding for these, to each other. We're going to put the alleles in trans to each other. Who's had organic chemistry? What's trans mean in chemistry? Flipped, right? On opposite, it means two substituents are on opposite sides of a double bond. And you can't rotate around a double bond. I know, chemistry, right? But it all, so I'm just telling you, it's all to get like physics, chemistry, biology, that's all the same thing. There's just lines drawn at levels of detail. Yeah. Yes. On the same gene. Let's just say that your gene is like a bicycle, right? Okay? And one bicycle has a bent wheel, so it won't work, right? The other, so you know that gene is defective, so when it makes the wheel, the wheel doesn't work and the bicycle won't work. The other one is missing the chain, right? So this bike has a bad wheel, and this bike is this gene makes the same bike, but it's missing a chain. Yeah. They are the same locus. There's two mutations. Let's say that the gene makes an entire bicycle. You know, I know. So then when you put them together, here's the one. This allele makes a bicycle with a wheel that doesn't work. This one makes a bicycle with a bad chain. They're both the bicycle gene. They're broken in different parts. So neither of them works. All right, here, let me do this. Right? So here's a gene, start codon, stop codon, right? One, and it all has to be perfect to code for the protein. Here's a gene. Oh, I was already talking. So this one has, there should be a T base there, and it's a C instead. Well, that would be a U. I'm just going to do this. So we're in the DNA now, so it would be a T, not a U. I don't want to confuse you. All right, so that's mutant number 1. You make that change and the protein doesn't work. Here is another allele of that in a totally different organism. And it's got an A to G mutation there. If you change the T to a C, the protein doesn't work when it's translated from RNA to amino acid sequence. When you change the A to a G, likewise it doesn't work. These are two mutant alleles in the same gene that give the same mutant phenotype. In other words, the gene doesn't work, but they're not the same allele. They're different changes. Does that help? No? Yes?
So when you say that, say the bottom one, if the bottom one was correct, I could put them together and they would complete each other because this one has all the stuff that I need for the top one doesn't. So
Hold on. Let me do this. Those that I drew are both mutations. The correct sequence is at the bottom. The top one has a C instead of the T, so it's broken. The bottom one has a G instead of an A, so it's broken. If I have this, let me erase this line now. If I have this as the genotype of one individual, it will not work. The top one has an A here, and the bottom one has a T there, like they're supposed to, but the top one, it doesn't matter that the A is correct because every other nucleotide is correct except for the C, but the C is enough to make it so that this one does not work, right? That will not make a functional protein. This will not make a functional protein. The cell cannot take pieces and parts of two genes and splice them together or splice the RNA together to make it work. Does that make sense? Everybody get that? So both of these genes, gene 1 and gene 2, are defective. They're just broken in different ways. They're different mutant alleles. Good, good? All right. Now let's go back here and give it another shot. So same gene, but this is one of the mutations we just talked about, the T becoming a C. This one is the A becoming a G, right? Mutant phenotype here because it's homozygous, mutant phenotype here because it's homozygous. You cross them together and you still get a mutant phenotype because you have what I showed on the iPad just now. Both of these genes are defective. It's just they have different mistakes in them. But neither of them will make a protein that works or does the job. Questions? I mean, I'm judging by the looks. You're like, mm-hmm. So, all right. So when you do this, you're putting the alleles in trans to one another. And so trans in organic chemistry means on the different sides of a double bond. Here, trans means one on one chromosome and one on the other chromosome. So this only works for diploid organisms, but because we're diploid, the world revolves around us, right? And other diploid organisms. So most of the organisms we use for genetics are diploid. So in this case, these fail to complement one another. This chromosome down here cannot complement the other one. So complement is total it out, right? So the other thing is like, like if you have a couple and the people are different, right? One of them is the more social outgoing one and one of them isn't. But the social outgoing one complements the other one and does most of the talking. And then the other one has some other feature that the first one doesn't. Here, you can't have the bottom chromosome complementing the top one or the top complementing the bottom one. They both have a mistake, and so you'd show the mutant phenotype in both. Good? All right. Right. Like two really, it'd be like me and another introverted person at a party like hiding in the corner together versus if you have one introverted person and one gregarious person and that person drags the other one around the room, glad handing everybody and smiling and all that stuff. So these fail to complement each other. So you need to know that term. When two alleles fail to complement one another, they are alleles of the same gene because they fail to complement each other. All right, let's look at the other example. I should also point out that in order to do a complementation test, first you should probably figure out if the two genes are on the same chromosome or not, because if the two genes are not even on the same chromosome, they cannot be alleles of the same gene because they're at different loci. So now we have gene A and gene B, like they're different genes. You can see that the A locus is over here on the left, the B locus is over on the right. So you still have an A mutant crossed to a B mutant, but in this case, even though they give the same phenotype, they're not in the same gene. I'm telling you that. So here you have a mutant over mutant for A, so you see the mutant phenotype. Here you have mutant over mutant for B. You see the mutant phenotype there as well. But because each of these is only mutant for one gene, there's only one mutant allele, the other one, the B here and the A over here, are wild type, so plus is normal. Good so far? When you cross these guys together, they do complement because the gene with the mutant A has a good B, which complements the defect over here. The gene that has the mutant with B down here has a good A, so it complements that one. So A and B complement each other, and because they complement each other, and when you have A over B, you get a wild type phenotype, then they must be alleles of different genes. This is called a transheterozygote because it's got two mutant alleles and two wild type alleles, but the mutants are in trans to one another. One mutant is on the top chromosome, that's the A. One mutant is on the bottom chromosome, that's the B. Good? All right. So here's the deal. When you're doing the point of doing a complementation test is that you don't know what I just gave you. You do a complementation test to find out whether two alleles are alleles of the same gene or not. And so you have to know what would happen if they are, previous slide, and what would happen if they are not, this one. So you do the crosses between the two mutants, get the heterozygote, and then find out, does it show the mutant phenotype or not? If it shows the mutant phenotype, then you know the alleles fail to complement each other and are alleles of the same gene. If it doesn't show the mutant phenotype, then the mutant alleles do complement one another and are alleles of different genes. You're ready to try something? All right, let's do this. So something I threw together here. There's two genes. I'm telling you, it's given they're on the same chromosome. Gene W1 and gene W2. And both of these, the W is like for white, like for, like the white eyes, so I'm just doing that. But if you are wild type, you get pretty blue flowers, right? W1 and W2 genes that I'm giving you show duplicate recessive epistasis, right? Precursor 1 is colorless. W1 enzyme, which requires W1 plus, makes precursor 2 also colorless. W2 enzyme converts that into a blue pigment that you see in the flowers. So if either of these is W minus over W minus, then you're going to get white flowers like this. So both 1W1 plus and 1W2 plus, and I didn't superscript those, sorry, are required to see the blue color. So here you're homozygous dominant for both of these, or wild type. Here you are recessive homozygote for W1, and so the flowers are white. Here you are recessive homozygous for W2, and the flowers are white. Everybody with me so far? So you have W1 and W2 genes. If either W1 or W2 is homozygous minus, you get white flowers. But they are, I'm telling you, W1 and W2 are different genes. They're not the same. All right. You're working at the greenhouse, and you come up with, you're working with the blue flowers, and independently you come up with 3 different new mutants. They're recessive, just like W1 and W2 mutants. They're A, B, and C. Each of these is a different mutant. How would you use a complementation test to determine whether each one of these is an allele of the W1 gene, an allele of the W2 gene, or something different? If it's not an allele of W1 and it's not an allele of W2, then it has to be an allele of some new gene that you don't know about yet. And that would be cool. Right? So let's just talk about mutant A. So I'm giving you homozygous blue flowered plants. Those would be W1 plus W2 plus. I'm giving you homozygote white that's W1 minus, and I'm giving you homozygous white that is W2 minus. Good? And also, you're getting homozygous, let's just do at mutant A, you're doing homozygous mutant A minus. So we need to find out if mutant A allele is an allele of the W1 gene, the W2 gene, or neither of them. Yeah. Oh yeah, you have to do that. Yeah. That is the complementation test. Yeah. So what would you do first? Pick one. So you're going to cross a mutant A homozygote with what? Okay. You said W1, right? Or 2? Okay. All right. So I'm writing this because the new mutant that's A minus, by definition has to be W2 plus because you're only mutant for one thing, right? So we're going to do this, and down here you're going to end up with A minus. Well, okay, so I can't tell you what it would be because I'm not telling you yet. So what happens is you would get, let's say, all the progeny have a white phenotype. So what just happened? They failed to complement and therefore are alleles of the same gene. Now here, in this case, this isn't exactly right then because I'm drawing them as 2 genes, but it's just the potential. So if that's white, then you have to like do this and this, and what ends up happening is the cross, this will actually be like that, and then the heterozygote, because they're the same gene, right, would be this. So at the same spot, W2 is over toward the right. Just pretend it's more over, it's more over to the right. So now I've shown that mutant A is an allele of W2. Really, if you did the work right, you could stop there. Why would you not have to do a complementation test to W1? Because we know it's an allele of W2. You can't have, an allele can't be an allele of two different genes at two different loci, and we know W1 and W2 are at different spots on the chromosome. Good, good? All right, so let's do mutant B now. Right? We're going to do this cross. And you get blue flowers. So what just happened? They did complement each other, and their alleles. So if they're on the same locus, this is what happens. Right? You get fail to complement because they're both mutant alleles of the same gene, but they're not. What happened? If they complement, then they must be alleles of different genes. And this is where, so again, try your very, very best to figure out what's actually going on and learn those concepts. Then you don't have to memorize, fail to complement and complement, different gene or the same gene, right? It's a 50-50 shot if you get freaked out on the test and forget. But if you know how it works, you don't have to have it memorized. You know. So in this case, it's some other thing. And when you get down here, your heterozygote would look like this. Because this chromosome is this one, and this chromosome is this one. That's not an A, it's just a little squiggly. Make sense? So now we know that B is not a W2 allele. What would you do next? You would do the same cross with W1, right? To see if it's that. Let's say that you got blue color with W1 as well. What does that mean? You cross B mutant to W1 mutant, and the heterozygote you get has blue flowers. Right, it's not that one either. So that would mean if you get blue flowers when you cross mutant B into both W1 and W2 mutants, that would mean that mutant B must be something else. And we don't know exactly where it is, but the gene would be like this. The B gene would be there, W1 would be here. We don't know where the B locus would be with relation to W1 or W2, but we know that it's not either of them. It is on the same chromosome, so it's somewhere else. And then we would have to do some genetic crosses to figure out where it is and what exactly it ends up being. Make sense? So you would do that with all of them, like with all of your new alleles, and figure out what's going on. you cross mutant C into you cross mutant C homozygote into a W2 mutant and get blue flowers. You then cross it into W1 and get white flowers. What does that tell you? And explain why. Right. Or so there's two ways. I could tell you what was done and what the results were and ask you what it means. Or I could do exactly like, let me stop this, do exactly like what's shown here. It's slow. Exactly what's shown here, and and say, you're given mutant B, and I would probably not do this. I would say, you're working with a white mutant in a flower, and you discover a new white mutant. How would you determine if the new mutant is an allele of the first one or something different? And then you would have to describe what a complementation test is, say, here, I'm not telling you what happens, so either it's going to be alleles of the same gene or it's going to be alleles of different genes. What would happen in either case? Then you do the experiment and see which one it looks like, right? We do this all the time in my lab. I do mutants that result in fertilization not working in the worms, so the worms are sterile. But there's like 60-some genes now that if you mutate them, they give the same phenotype. The worms don't lay eggs, they just lay oocytes. So when I get a new mutant, the first thing in a screen, the first thing I do is I have to find out what chromosome it's on. Then I have to try it in complementation tests with every doggone mutant that's already known on that chromosome. Because the work to find out what a new gene that I've gotten in a mutagenesis screen is a lot of work. And you don't want to do that work only to find out that your new gene is something somebody's already published and you've got to throw it in the trash can. Either way, it goes in the trash can, metaphorically, but you can do complementation tests at first and cut off all that extra work at the end. So these are actually, this is actually widely used. Good? All right. Let's move on. So we're going to talk about sex-influenced characteristics and sex-limited characteristics. These are not sex-linked characteristics. Sex-influenced and sex-limited are autosomal. It says it here, but when you hear sex, you're going to think X and Y chromosomes, and it's not. So sex-influenced is an autosomal characteristic with higher penetrance in one sex versus another. Pretty much what it is an allele that is dominant in one sex and recessive in the other. The examples they give here is beards and goats. So B with a B is a beard. B with a plus is no beard. But the BB, the beard allele, is dominant in males but recessive in females. Because males have higher testosterone levels and other male stuff that's going on there. That's the difference. It's the same allele performs differently because a male and a female are different hormonal environments. So here we're going to have a cross, a beardless male and a bearded female. They're both homozygotes, so you end up with an F1 generation that is B plus and then BB. However, even though they have the identical genotype, males with this genotype will be bearded while females would not be bearded because it is a sex-influenced characteristic. So then you're going to do the F1 cross and into the F2 generation. Even though this female does not have a beard, she's still B plus BB, and so the underlying genetics works exactly the same. So all the F2 generation are going to have a 1 to 2 to 1 B plus homozygote, B plus BB het, BB BB homozygote genotype ratio. But it's going to be different in males versus females. In males, you're going to have 3/4 beards because all you need is 1 BB, so BB blank. In females, only 1/4 is going to have beards because the heterozygote in the female environment does not produce a beard. So the genotype and phenotype ratios change based on the sex in this case. Sex influenced. Sex limited means, oh whoops, I forgot I had this problem in here. All right, so coat color in this breed of cattle is a sex influenced trait, right? So what is the sex of a red calf? So calf is a generic non-sexed term. Born to a mahogany cow, right? So a cow is a female, the bull's the male. So in this case, you can see that there's a sex influence. So the M plus and the MR. The M plus is mahogany and the MR is red. But you can see that there's a difference in males, whereas in males, the M plus is dominant for mahogany. And you have to be MR-MR homozygote to be red. And in females, the MR is dominant to M plus. Just a difference in sex. So, what is the sex of a red calf born to a mahogany cow? So if the cow is mahogany, what does her genotype have to be? M plus, right? So, what does the genotype of the calf have to be? Well, it's got to have one M plus, right? Because the mom is a homozygote. The calf is red, so it's got to have at least one MR. So the genotype of the calf has to be M plus MR, and the calf is red, so what is the sex? Female, because that's the only thing that's possible there. Make sense? All right. A sex-limited characteristic, also autosomal, but is only possible in one sex. Here they have the spectacular long plumage on roosters versus this little, you know, the hen feathers, the hen bun, I guess, or whatever you would call it on the back there. So to have the plumage, you have to be homozygous for little h, little h. If you are male and you are big H blank, too bad, dude, you look like a hen. But in the female, it doesn't matter what you get, even if you get the alleles for the impressive tail feathers, because you are a female, you don't have something else in your genotype, and I don't know what it is, to get that. And so therefore, you have this. However, you are carrying these whatever alleles you have, right? So in order for the rooster to show this, the hen has to be at least one little h. So I guess you would say little h blank or blank little h, right? Because the male would have to be little h, little h. Considering that I rarely see, you know, in chicken pictures or whatever, people posting roosters that look like that, that's probably homozygous for this, and this is just a mutation, right? Yeah.
So, I've had chickens, and when you take away the females develop tail-like characteristics. Is that because of their hormonal environment? So they have the alleles to develop it, but
What male characteristics do they get?
they get larger combs, they behave like a male, they get a bigger tail. It's weird.
And it can change if you bring the male back, it shrinks back down. It probably is something along those lines, but not being allowed to have chickens myself, by spousal ordinance, not by city ordinance. I do not know the answer to that one. And that would make it so that if you take away the mix, see, that's what I said, most of them are, everybody's probably like this, because when do you see a rooster without the big tail? So they probably are all like that. It's just when you take away the rooster, it's probably a behavioral thing, like the brain changes a little bit or something. And I've actually seen people are complaining here and there because I'm not allowed to have chickens. I go on, like, I watch the chicken Facebook groups as sort of, and then like the female chickens will behave like the rooster and like, mounting type activity and stuff like that if the rooster's not there. So I know that happens. All right. See you guys on Tuesday. Sometime today I will get the next assignment posted on Blackboard. And I will see you guys at 2:10 today at the lab if you are on the Thursday lab. Remember, office hours are going to end about 11:50 today because I have to go to a meeting.